Thank you first for the invitation. So we come from a group that is in the Michaelis Institute, which is a large microbiology lab here in France, in the Paris region. And we have been doing some using and developing machine learning for quite some time. We started by doing things that are rather classical, where we acquired, let's say, training set, and we made some predictions on this training set, for instance, about promiscuity of proteins, and then we went on and did some validation, and discovering, for instance, new metabolites in E. coli. Then we also worked on things that are more entwined with synthetic biology, especially within the DBTL cycle, and we developed a method called active learning. In fact, we use it. It was developed by in other contexts, but we use it within the DBTL cycle to optimize some processes. But today I want to talk about something that is more exploratory, which is basically looking if we cannot use, rather than using in silico machine learning, if we could not use the biological substrate to perform the same task than you do with classical machine learning technique. And this is rooted on a perhaps naive analogy. You know, when you look at basically a neural network, for instance, you have a training set, and you're going to learn the weight of your network in order to be able to make some good predictions. Now, if you look at microorganisms, in particular prokaryote, where there is no much of signaling networks there, everything is happening through metabolism, through metabolic network, which basically are processing environmental signals, which are other metabolites, and transforming them, okay? Not with the goal of solving any problem, but with the goal of perhaps the best goal of all, to survive. And so the idea that we have been looking at was perhaps we can divert these metabolic networks in order to use these to also solve problems, and solve problems that are usually dealt with a neural network. So what I'm going to talk about is already a preprint that you can find on bioRxiv. So to start, of course, metabolism is for food processing, okay? And it is not really an information processing apparatus. It's not viewed like that, okay? But nonetheless, there are some evidences, you know, that metabolism is also playing a role in solving complex tasks. And for instance, one good example is the blob. This is a unicellular protist that is finding its way through a maze by producing and detecting molecules. Okay. Another example is with plants, where you have several layers of production of metabolite and also signaling pathways, which has been termed the plant perceptron. And finally, if you look at it, if you think about it, the neurotransmitters that we are producing in our neurons and that are activating chemical synapses are also metabolites, and actually central metabolites. So here again, metabolism is playing a role into these complex tasks. And finally, also a reason why we move into this is because in the past, a couple of years ago, we built a metabolic perceptron. It was done in cell-free system that enabled us to classify samples based on their metabolic composition. So a perceptron, and that's what is shown here on the left side, is a simple weighted sum, you know, of some input signals, okay? And if you want to do a classification, usually you add to this an activation function that is a sigmoid. There are other activation functions, but sigmoid is good for classification. So the way this is working is, first we train this in silico perceptron in silico, okay, for an arbitrary classification problem. Okay, some cases are negative, some cases are positive. Then the way we execute the perceptron in vitro, in cell-free, is that we instantiate the signal by different metabolites, okay, that are transformed by enzymatic transformation into an activator of a transcription factor. And downstream of that, we can express a fluorescent protein. So it's fairly easy to get the weight, because we need to do a weighted sum into the system by just controlling the DNA enzyme concentration. So, and at the end, it worked quite well compared to what was expected. So the next question is obviously to do this in vivo, because it was done in cell-free system, and it's rather difficult, you know, to fine-tune the expression level of enzymes in order to implement the perceptrons. But perhaps we don't need to do that. We just maybe can work directly with the metabolic network of wild-type strain. And so the proposal is as follows. You start with a machine learning problem, and the instances are all the time described by features. And you are going to transform these features into nutrient concentrations that you're going to add to your growth medium. Then you have your strain, you're going to grow your strain, and you're going to measure the phenotypes. Okay, so the phenotypes can be, for instance, the growth rate, can be the oxygen consumption, or anything you can measure, including even biosensors that are measuring the level of different internal metabolites. But once you have these phenotype measurements, then you feed a second neural network, which take these as input, and finally print out the solution to your problem. Okay, so that's the general idea. But in order to do this, you need to be able to backpropagate the gradient from the post-ANN to the prior ANN. Okay, and in order to do this, you need a model that accurately represents your strain and that also enable to pass the gradient through its operation. So one problem that we have with metabolism is when we talk about genome scale models. Okay, the technique to study the dynamic of a metabolic network does not allow to backpropagate the gradient. But so we looked at what was happening in other fields outside metabolism, and as it turned out, in the signaling network, people have also looked at this issue of replacing classical neural network by signaling network, and by basically, instead of having, say, a classical neural network with dense layers, by replacing the connectivity of the layers with the actual connectivity between the proteins that you are going to find in your signaling network. So every node here and every edge is something that do exist in the signaling network. And they have shown that when they train the network, by looking at the weight, they can find out in the T-cell receptor signaling network, they can find out what are the important protein and connections in this signaling network. More recently, people have recognized that in signaling network, you have loops, okay? So you cannot only use feed-forward network. So you need to use a recurrent neural network. But here again, you know, the connectivity between the nodes correspond to the connectivity of the signaling network. And once you train this, and they have done this on training with different ligands that activate transcription factors, once you train this system, you obtain fairly good correlation coefficient. This is the R-square. But what's really important, and I'm saying this because I'm going to use these terms later, is to look what's happened in instances when you make prediction on instances that have not been seen during training. And as you can see here, the value are getting quite lower in this case. So anyway, so can we use this idea for metabolic networks? So what I'm showing here is a snapshot of the upper glycolysis pathway of E. coli. Okay, so this is a part basically of a metabolic network. So typically, what you do when you study the dynamics at genome scale, you use this so-called flux balance analysis. Okay, where you give as input the fluxes of the metabolite that are entering the cell, the intake fluxes, and you maximize some objectives. So usually the objective that you maximize is the growth rate. So here that would be the rate of the biomass reaction. But doing that, you have some constraint. And the constraint is that at steady state, the equilibrium, the production of the metabolites, the flux of production of metabolites equal the flux of consumption of metabolites. And that is captured by this equation, SV equals 0, where V is the set of fluxes. And so anyway, so you can solve this using a simplex algorithm. This is a linear program, and you get the steady state solution. So what's interesting is to look at some relationship that you have at steady state. For instance, you have different matrices like the flux to metabolite matrix and the metabolite to flux matrix. And you also have a weight matrix that basically allow you to compute the fluxes of the reaction from the fluxes of productions of metabolites. And we're going to use this relationship later. So let's take the signaling network idea, and let's start by developing our network. So first, we know that we have a flux entering the cell. Okay, so V1 is going to populate M1, and M1, this metabolite, is contributing to two different reactions, V2 and V3. So a fraction of M1 is going to go to V2, and another fraction is going to go to V3, the sum of the fractions being equal to 1. And then you iterate this process, okay? And then M1, M2, and M3 get populated, and here again, M2 is split between two reactions. So you have two additional weights. And you iterate this process until all the nodes are populated and until the values of the M and the V's get stabilized, okay? So this is nothing else but a recurrent neural network, okay? Where you update at each time you go through the loop, the value, the production flux of the metabolites and the reaction fluxes. And this is easy to simulate. And what you see here is a simulation of that. And it requires only 30 steps to get to the value. And that's, of course, the same value that you would get by using the simplex algorithm. So it's all great, and it seems that our problem is solved. Well, not quite, okay? So if you look at this network, this is the same network, okay? But we have different intake fluxes, okay? 1 and 0.5 in case of A, and 0.75 and 0.25 in case of B, okay? And in both cases, the steady state fluxes satisfy the constraint SV equals 0, okay? But now if you look more closely at this, you find out that the weight are different. And the reason is that there are two substrates in this reaction, okay? And the flux of that reaction equal the minimum entry flux, okay? And so that means that in this case, W41 equal half, and the same for W31. But in that case, okay, it's only 0.25, and it's therefore 2/3 and 1/3. Okay, so all of that means it's bad news. It means we cannot find a set of weights that will satisfy our network because the weights are depending on the input fluxes. Okay, so that kind of invalidates this simple idea of having a weighted network like we had with signaling networks. Okay, so we kind of got disappointed when we found this out. We first tried to have a layer that would take the weights, but we also look at the literature and found out that, in fact, a linear program and a quadratic program also as well had been solved using a neural network and recurrent neural network in the past, in the math literature, okay? And we ended up choosing two different solutions. One where we provide exact bounds for the intake fluxes, and one where we provide upper bound for the intake fluxes. And so we encoded this, and as you can see here, you get in fact a perfect result, and the recurrent neural network mimic exactly what would be given by a FBA simulation. And we did this on only different growth rate that were generated by CobraPy, software to study the fluxes, on E. coli core model, where we varied the concentration of 13 different medium, mostly these were sugars. And so we do have a perfect correlation for both the upper bound and exact bound, okay? And but one caveat is the number of iterations. As you can see here, the number of iterations is quite large, okay, almost 1 million, in order to reach the actual value. So one way to look at this was to look at when we started to introduce some noises, you know, in the solutions. And you can see that once you start to do this, you get much less iteration, you know, in the process in order to converge. So basically, the idea of our artificial metabolic network, okay, is to have a first dense layer, okay, that which purpose is to compute this initial vector of fluxes that is not too far from the solution, okay, and then using a mechanistic layer with this RNN cell that I showed earlier to calculate the final fluxes. And so this is what we implemented. And as you can see, we have a very good matching as far as training and also validation set is concerned, okay? And this is compared with what you would get if you had a simple dense layer just that would do a very basic learning on neural network. And you also validate the constraint of SV equals 0. So this was done on an E. coli core, which is a small model of E. coli, and IML 1515, which is the full E. coli model. So having done this, so what we wanted to do was, and that's what's also interesting with this approach, is that we can train not only on simulated data, FDA simulated data, but we can also train this on experimental data. So for this, we generated 73 different media composition. They were all composed of M9 plus glycerol and 27 extra metabolites that we added to the growth medium. So we actually added between 1 and 5 different metabolites that were chosen at random in each medium. And all metabolites were added with the same concentration. So mostly they were sugars and amino acids. And what you can see here, and again, it's compared with a dense network, okay, that the network performed quite well, okay, and with a Q square, okay, of 0.72, which is done really on the data that had not been seen during training. So all of this is encouraging, okay? So what we looked at later was to predict medium intake fluxes with AMN, and for this, because we had only concentrations. So in order to transform the concentrations into the fluxes of entry, we used the train AMN that was trained on Cobra Pi results, okay, and we transformed the concentrations into intake fluxes, okay. So the intake fluxes, unfortunately, are not known for the metabolite entering E. coli cell except for few ones, okay. And so for this, we use this network here as a non-trainable network. This is what it's called a reservoir. So it was not trained anymore, okay? What we trained was the fully connected dense layer, which purpose was just to transform the concentrations into values for flux getting in, okay? And what you can see here again is that we get fairly good correlation coefficient, okay, even in validation in a test set, okay, that were not seen during training. Here we use the leave one out procedure, and we got also, of course, a fairly good regression coefficient. So now it's interesting because we know what are the intake fluxes, okay, going into our E. coli model, and we can actually use that data and directly use the flux balance analysis, okay, and you get by taking the values, you get using only flux balance analysis here, you get a very good correlation coefficient. This is to be compared of what you would get if you were going to use straight the flux balance analysis by saying, okay, if my metabolite is present in my growth medium, I'm going to give an arbitrary value of 10. Okay, if it's not present, it's going to be 0. Then you would get fairly poor result as far as a correlation coefficient is concerned. You would get a little better if you start to have some optimization where you try to optimize the intake fluxes in order to minimize the error between the measured growth and the predicted growth. But anyway, here it seems, at least that's the first application of this artificial metabolic network, is to provide the right intake fluxes that you would have in your model when you run the flux balance analysis. So I am done. And wanted to thank you, 3 grad students that worked on this. Léon Ford that actually generated the data, and Bastien and Emmerich that work on different parts of the code, along with some scholarship for Léon from INRAE, and a newly funded project to work on this artificial metabolic network that we got from the French equivalent of NSF for the ANR.